Okay, thank you for the introduction. And of course, I'm going to talk about trauma generation. My name is Bas de Laat, and I'm the General Director of Synapse, the organizer of this conference. We just had a little introduction by myself and Mark, but now we'll go into much deeper into trauma generation and Synapse, and especially what Synapse did over the last already 22 years that we are busy with trauma generation. So, the Synapse Research Institute. So, we were founded in 1999 as a spin-off from the Maastricht University. In 2009, we became independent from the university. And when we moved to the Lambertus Church in Maastricht, we were also officially recognized by the government as a not-for-profit research institute with especially great social impact. Our core activity is to explore trauma generation, as many people know. But we also have other activities like platelet function, fibrinolysis, and we also have clinically oriented research. We have many collaborations with other medical hospitals, also laboratory service to pharmaceutical companies. And one of the last things that we also do is we are specialized in binding characteristics as we bought Delbia a few years ago and use ellipsometry. And when Synapse was found in 1999, it was found by Professor Hemker, the former rector magnificus from the Maastricht University. He didn't want to retire. He wanted to go on with research, and it was a good decision because from there on, trauma generation got really a boost due to his activities in this field. Here you see a slide where, we obtained Delbia to include protein binding to its portfolio. And why do I show this slide? Because both trauma generation is very important, but also the binding characteristics of proteins are very important. And especially because the first thesis defense in Maastricht in 1976 was based on the subject of ellipsometry, and it's especially very important for Synapse because the promoter was Professor Hemker, and it was the first thesis defense in Maastricht. So back to trauma generation, our core business. So you have trauma generation in plasma, you have platelet-rich plasma, and you also can do trauma generation in whole blood. And let's start with platelet-poor plasma. So the plasma trauma generation, it is not new, and it was also not founded by the people of Synapse, as it was already published in 1952 by Professor McFarlane, a trauma generation test. And if you look into the publication, you really see trauma generation curves. And it was published as the application in hemophilia and thrombocytopenia. And then Professor Hemker and Professor Béguin continued and initially performed trauma generation via the subsampling method using a chromogenic substrate. Of course, you get really nice trauma generation curves, but this required expert laboratory skills. And also 2 persons are needed to perform the assay. One that can pipette, the other one records the time to stop the reaction. It was quite labor intensive. Then we came to the CAT-based trauma generation. This all started at the Department of Biochemistry at the Maastricht University. As you can see in the upper picture, Professor Hemker is busy and many people watch as trauma generation gave also not only a boost to Synapse, but also to the Department of Biochemistry. And after it was founded in 1999, we moved to the Biopartner building, as you see in the picture below. It continued in this building. First, it started with 2 laboratories, then we got 4 laboratories. Then at the peak, we had 7 laboratories that were busy with trauma generation for Synapse. And then we grew out of the building. So in 2016, we moved to the former Lambertus Church. And over there, we have now a big laboratory with over 1,000 square meters of lab and offices. And at the moment, 38 people are working at Synapse or in laboratories associated with CNAPS abroad. So the CAT-based trauma generation. So we had Synapse, but Synapse at that time still Synapse BV, a company, wanted to do research, and then Trombinoscope was founded, and Dr. Giese, he continued with Trombinoscope to do the marketing and the selling of the machine. And over here you can see the Thermo Fisher machine. Many people use this machine, and it proved to be quite a success. And for this thrombin generation, this calibrated method, this requires the simultaneous measurement in two wells. So you have a reaction mix and a calibration mix. So the reaction mix is the plasma that you want to test. You have a trigger, of course the substrate and the calcium by which the measurement starts. And with the calibration, you also have the plasma, but you have a calibrator, that's alpha-2M thrombin, and then you add the substrate and the calcium, and then also that reaction starts. Here you can see the continuous measurement of thrombin. You can see in the black line, you see the calibrator, and then the calibrator can be corrected to a straight line. And you also see in gray, you see the thrombin generation and the thrombin generation corrected. In the right graph, you see the thrombin generation curve, as many people know. You also see the dotted line, and the dotted line is not corrected for the alpha-2M activity, because we found out that our substrate still can activate, our substrate can still be activated by thrombin, although it's inhibited by alpha-2M. So we did a mathematical correction for this, so that you have a thrombin generation curve that also goes to 0. This thrombin generation method, that made really thrombin generation famous in the world. People started to use it. It's user-friendly, especially in the research. And here you can see you can measure many things and the effects on coagulation, which is not possible with, for example, a PT or an APTT. We can measure with thrombin generation on a very sensitive level. You can see over here in this control, that's a normal generation, thrombin generation curve. You can see the rivaroxaban, for example, that has a flattened curve. You see the effect of dabigatran on the plasma. It's a prolonged lag time, and you can see the curve of enoxaparin. It's also a little bit prolonged lag time and also a little bit flattened curve. What you also can see is that these anticoagulants have a very differential effect on thrombin generation, and also on the basis of the curve, you can identify which anticoagulant is present in the plasma. Well, all congenital acquired thrombotic tendencies can be measured by TG because it goes up. And one of the most used tests that can be applied to thrombin generation is the addition of thrombomodulin or APC. Because with this test, you can measure the, for example, the factor V Leiden, so the activated protein C resistance. You can see in this curve with the blue curve, you see, addition of thrombomodulin. Then you can see in the green curve, you see the addition of activated protein C. Then you see in the red curve thrombomodulin. You can see that both thrombomodulin and APC inhibit factor V, which then decreases the thrombin generation curve. And with factor V Leiden, this is not the case. Now, of course, thrombin generation has been tested in many other different diseases, and here you can see a whole bunch of publications, quite old publications, but still very valid, where thrombin generation was increased, and when thrombin generation is increased, there is a higher incidence of venous thrombosis. Then also thrombin generation can detect congenital or acquired bleeding tendencies, and then thrombin generation is decreased. And what has been shown is that all congenital deficiencies causing an ETP lower than 20% have a bleeding tendency. In this slide, you see the effect of a factor V deficiency on thrombin generation, but you can do this for many other factor deficiencies. You can see when factor V decreases to really low concentrations, this has a tremendous effect on thrombin generation. So, was this the right time to start measuring TG? Yes, but I think many people also know the difficulties that we encounter when we're doing thrombin generation via the CAT method. Because the CAT method is not CE marked or FDA approved. We found that when you do one thrombin generation in one laboratory, still when you measure the same sample in another laboratory, you have different results, and even different patient populations give different results. And most things come out of this method, this thrombin generation calibrated method, is not optimized. So that's why Stargo introduced several years ago already the Genesia, and the Genesia is also a method to measure thrombin generation, but it's a different method of measuring thrombin generation, but it's fully automatized. And it was introduced over here in our laboratory by Professor Hemker. And now I will show a movie where you can see the methods of measurements of the Genesia. After thrombin generation in plasma, we started to do thrombin generation in whole blood. Over here you can see the slide of the evolution, how we see it, of coagulation testing. First, of course, we had the normal coagulation tests, the clotting tests like the PT, the APTT, the DRVVT, and other clotting tests. Then we started to do thrombin generation, very sensitively measure coagulation like the former tests, only we can measure prothrombotic phenotype which is almost impossible with the coagulation tests like the PT and APTT. And then we started the whole blood thrombin generation. So why whole blood thrombin generation? Well, plasma thrombin generation, and there we add artificial phospholipids, and with whole blood thrombin generation, we add the real cells. And you can see over here in this slide that many coagulation proteins are dependent on phospholipids, and we think that using the natural phospholipids is a more physiological way how to measure coagulation. So in 2012, we published the first attempt to measure whole blood thrombin generation on a thin filter paper, and it really worked. We had a thin filter paper that we added to a 96 well plate. Over there we added whole blood and some oil, and then we measured thrombin generation. But there were also some disadvantages to the whole blood CAT paper method. Really extensive training is required. I think there were only 3 or 4, maybe 5 labs that were able to do it. You have to be really fast. And we use a porous matrix. So we introduced the foreign material into the blood. And we also found out, maybe due to this porous matrix, that the test is independent of platelet number or function. So improvements were required. So in 2020, we developed a new method for measuring thrombin generation in whole blood. Here you can see the conditions, how we do it. But the most important thing is, from the slide, is we do not use any foreign material that we have to add. It's just a normal CAT method that we use also in the CAT machine. And we can even use sometimes with certain samples the normal software to measure thrombin generation in the whole blood. Most important thing is the difference between the plasma and the whole blood is that we do continuous mixing, because you have sedimentation of the red blood cells. Here you can see that we did a comparison between the old and the new methods, and especially if you look at the time-dependent parameters as displayed in this graph, you can see that the lag time is really longer when we use the new methods. So on the x-axis it's called no paper, than with the old method where we had the porous matrix, which is called with paper. And also time to peak, you see it's way longer. So we thought that there's less contact activation compared to the whole blood CAT method when this was on a paper matrix. You can see that both with 0 tissue factor and 1 picomole of tissue factor on the right, we see that the method that doesn't use any paper has a longer time-dependent parameters, which indicates that there's less contact activation. So the conclusion is that the whole blood thrombin generation on the CAT gives a good reproducibility. It is tissue factor dependent. The test, as shown in our publication, is sensitive to blood cell numbers. The test is sensitive to platelet function, and there is no need for a foreign matrix to prevent sedimentation. So big improvement, and this method is better to introduce into other laboratories on a research basis. But also this whole blood thrombin generation makes other studies, novel studies, possible, because you don't have to spin down the blood. And also on the basis of this method, we developed a really prototype, not ready for market, point of care method that we can use anywhere. And that's made our hypoxia studies possible. So we went 4 times to the Swiss Alps and 2 times to the hypoxia chamber, where we investigated the effect of a situation where there's less oxygen on different tissues of the human body. And we hope with these studies that we can translate this into people that have less oxygen in certain organs or people with pulmonary problems. And what we've seen so far with these studies is that hypoxia introduces a prothrombotic phenotype, and that the effect on platelets differ from that on the plasmatic portion of the blood, and that there is a difference between men and women. And to give an indication of how those studies look like, Here's a short movie of our first expedition that we did to the Swiss Alps. And now, so now we come to different parts. I've shown plasma thrombin generation as it happens in our laboratory, whole blood thrombin generation, how we do it in our laboratory. But we also have thrombin dynamics. And what is thrombin dynamics? With thrombin dynamics, we study the balance between the pro and anticoagulant processes. Here you can see a thrombin generation curve on the left, which is a normal thrombin generation curve by which the prothrombin conversion and the thrombin inactivation are balanced. But when there's a disbalance, for example, when the thrombin inactivation is larger than the prothrombin conversion, the thrombin generation curve is lower. But also when, for example, the prothrombin conversion is increased and the thrombin inactivation stays the same, then your thrombin generation curve is increased. And with thrombin dynamics, we get way more insight into the details of a thrombin generation curve. And we can better study the balance between the pro and anticoagulant processes. Here you can see again, thrombin generation is the result of the prothrombin conversion and the thrombin inactivation. Here you can see in this slide how we measure thrombin inactivation. By that, we do a known amount of thrombin into the plasma and look how the fluorescent signal decreases over time. And by that, we see how fast thrombin is inhibited by antithrombin and all the other inhibitors. Then we compute the thrombin inactivation. And this is a result of many years of research, how we looked how antithrombin can inhibit thrombin, how fibrinogen has an effect on the inhibition of thrombin, and also alpha-2m has an effect on inhibition of thrombin. Then we calculate the prothrombin conversion. And like I said before, again, over here you can see the figure that I've shown before. Thrombin generation is the result of prothrombin conversion and thrombin inactivation. Here you can see a typical prothrombin conversion curve. It's sort of a thrombin generation curve, but that only for prothrombin conversion. And in the bottom you can see the equations that we use to calculate this prothrombin conversion. I won't go into detail, but you can see that we have constants for antithrombin, for alpha-2m to calculate the inhibition of this thrombin. Here you can see which parameters that we use for the thrombin dynamics. For the prothrombin conversion, you can see again a prothrombin conversion curve. We use the maximum prothrombin conversion rate, but also the total prothrombin that's converted into thrombin. And for the thrombin inactivation, we can calculate the thrombin decay constant. So for which clinical diseases have we used it? Well, one of the best examples is liver cirrhosis. And liver cirrhosis is a very difficult disease for hematologists and people that are studying this liver cirrhosis and thrombosis. because there's no clear bleeding or thrombotic phenotype. Sometimes people bleed and sometimes people have thrombosis. There's an increased risk of both types of hemostatic events, but also the coagulation factor levels are reduced due to the liver function. And here you can see, for example, a thrombin generation curve. They look a little bit the same. You have the healthy thrombin generation curve, and you have the child group A, B, and C. And from A to C is an increase in liver cirrhosis. You can see that are quite the same. So on the basis of a thrombin generation curve, I cannot say who's going to bleed and who's going to get thrombosis. But then we went into detail. So we did the thrombodynamics on this liver cirrhosis patient population and calculated prothrombin conversion. What you can see also is the prothrombin levels, they decreased with an increase of liver cirrhosis. But you can also see that the prothrombin conversion increases with an increase of liver cirrhosis. So that's contradictory. But if you then look at the total prothrombin conversion from child group A to child group C, you see a decrease in prothrombin conversion. So that looks like a bleeding phenotype. And you can also see with the maximum prothrombin conversion rate that with increase of liver cirrhosis, there's an increase in risk. So there are many factors that determine a thrombin generation curve and that also determine your bleeding or thrombotic risk. We also, for example, measure the antithrombin levels. Here you can see that the antithrombin levels, so the inhibitor of thrombin, is decreased with increasing severity of liver cirrhosis. So many things happen with liver cirrhosis. And we think that with thrombin dynamics, we have way more insight into the clinical phenotype of a patient with liver cirrhosis. Also here, the next graph, you see the thrombin decay capacity. And with the antithrombin levels that decrease, you can also see that the thrombin decay capacity, so how good thrombin can be inhibited, decreases with an increase of liver cirrhosis severity. So then the last part that I would like to show what we do with thrombin generation within CNOPS, and that is artificial intelligence. Artificial intelligence is very abstract, so I will try to explain it very slowly so that, that people understand what we do. It is not that difficult, only you have to know what you do. And as an example, I will use the antiphospholipid syndrome where we show thrombin generation and artificial intelligence and how they can help each other to diagnose a patient with the antiphospholipid syndrome. So the antiphospholipid syndrome is an autoimmune disease. People have antiphospholipid antibodies and they have thrombosis and pregnancy mobility. And why is it a syndrome? At this moment, we still don't know exactly how these antiphospholipid antibodies induce thrombosis or pregnancy mobility. And what we now did is, in this patient population, we also use thrombin generation besides the classical antiphospholipid tests like lupus anticoagulant and the anti-beta-2 glycoprotein-1 antibodies and the anti-cardiolipin antibodies. Here you can see the principle of the neural networks. So you have a biological neuron, we all have. You have an input where the synapses are. You have a cell body where the processing is, and you have an axon that regulates the output. And that's also how we use it. Here you can see on the right an artificial neuron. You have the input, we put all kinds of parameters in it. Then you have the brains where it's calculated, and then you have the output that says, do you have are you diagnosed with the antiphospholipid syndrome or not? Here you can see the principle of a neural network. You can see here the input is a dog, then it goes through all the neurons and you can determine how many neurons you use, and the output says it's a dog. So very simple, so that's how we use it. So here you can see the thrombin generation, but also the dynamics of thrombin generation, how we calculate it. First, we wanted to see how a population of antiphospholipid patients looks like with thrombin generation. And you can see that there are differences. There are significant differences between the controls and the antiphospholipid patients. Like with the lag time, it's shorter lag time indicates a prothrombotic phenotype, which is true for the antiphospholipid syndrome. You see the peak is higher, the time to peak is shorter, the ETP is a little bit lower, that's strange, but also for the thrombodynamics, we see that the total prothrombin conversion is higher, the prothrombin conversion, maximum prothrombin conversion is higher, and the decay is lower. So everything shows that there's a prothrombotic phenotype. But you can also see the problem. On this data, we cannot diagnose a patient that having antiphospholipid syndrome or having thrombosis because the populations really overlap. So that's why we need artificial intelligence to see whether a computer can find unique combinations of tests to diagnose a patient with the antiphospholipid syndrome based on our data. So we developed a neural network. So we had a patient population of 31 APS patients and 66 controls. We had a neural network where we did an input, the thrombin generation parameters, the lag time, the peak, the ETP, the time to peak, and also the effect of thrombomodulin, but also the parameters of thrombin dynamics, like the PC tot, the PC max, and the thrombin decay capacity. And our output should be an APS patient or a control. And then we had a clinical validation cohort, of course, to see whether our artificial network that we constructed on the first population also works on other populations. Here you can see the results of our diagnostic neural network. And we had a quite good accuracy of 99%. And you can see that the positive predictive value is 100%, and the negative predictive value is 98%. So by using artificial intelligence and data that seems not to correctly differentiate between patient samples and controls, we were able to construct a neural network that was able to depict an antiphospholipid patient based on its clotting status. Here you can see the relative importance of the individual parameters. And here you can see that the thrombin decay capacity was the most important factor in diagnosing a patient with the antiphospholipid syndrome. And then everything is ranked by importance. On the basis of these parameters, we were able to diagnose antiphospholipid patients without any of the classical other diagnostic criteria like the anti-beta2 glycoprotein 1 antibodies, the lupus anticoagulant, or the anti-cardiolipin antibodies. So with that, my presentation comes to an end. I would like to acknowledge every center that we work with over the last 22 years. And there are a lot, so I won't mention them because then I will forget one. But I would like to have a special thanks to all the employees of CNAPS over the last 22 years. With these people, we have built a really strong institute that can hopefully exist for another 22 years. Thank you for your attention.